This lecture talks about host microbe interactions and epidemiology. Host microbe is talking about the host, the human host, and the microbe or the microorganism that's going to live in or on that human host. And then epidemiology is the study of diseases in large populations. So a patient comes into your clinic with C. diff, short for Clostridium difficile, an opportunistic pathogen that is highly resistant to antibiotics and causes severe and sometimes fatal diarrhea. A fecal transplant is discussed as an option for treatment. The patient freaks out. Fecal transplant? Why would you give me feces? What is in the feces that's beneficial? What is an opportunistic pathogen and how did I get colonized with this pathogen? So these questions are going to be explored in this lecture. So to begin, why are we giving this patient feces? Well, the patient likely had a prolonged course of antibiotics or something wiped out their normal bacteria, which allowed for the overgrowth of this C. diff. C. diff is an opportunistic pathogen. It usually only causes infections in people that are immunocompromised or somehow compromised, like their natural bacteria got wiped out. So one of the best treatments for C. diff is not going to be giving them antibiotics because they've likely already been on antibiotics for a period of time. It's you got to get the good guys back. And the richest source of the good guys is directly in feces. So usually they'll ask someone that resides with the patient to donate a fecal sample. And then they'll process the fecal sample and it goes through a tube up the nose and down into the stomach and dumps into the stomach so that it can colonize the remainder of the digestive tract. Sounds kind of gross, but it's a really good source of probiotics or the good guys, the good bacteria. And if you live with this person, chances are you share 99.9% of the same microbiota anyways. So just by that close association, you're getting colonized with stuff that you probably already had. Now, there were some clinical trials that were recently shut down involving fecal transplants here. So they tried to do these fecal transplants in clinical trials, and it ended up killing somebody because they ended up getting a severely resistant microorganism, an enteric pathogen. So they've suspended clinical trials temporarily, but this is something that can be done as a treatment option. So starting off with some definitions, a pathogen is a disease-causing microorganism. To know the difference between being colonized and being infected, you have to evaluate the extent of damage or no damage that's done by the microorganism. So when we say that a patient is infected, it usually means that there is a pathogen that's causing harm. So they're harboring some type of pathogen that is harmful, causing some type of damage. Colonized just means that the bacterium is in you or on you. It doesn't necessarily mean that it's causing damage. And a lot of times when we say a patient is colonized with something, it's usually a good thing. So they're colonized with good guys. Not all the time, because you can be colonized with a pathogen, but in general, infected means bad guys, colonized kind of means the good guys, or they're not really causing a disease state at that time. Pathology is the study of disease, and that's not just microbial diseases, it's any disease. So a pathologist would study heart disease or liver failure. Microbiota, resident flora, this is the normal bacteria that lives inside or on the surface of a person. So this is your natural resident or natural microorganisms that live in you or on you. Some of the stems that you may hear as we talk about these various diseases are derma. Derma is a disease of the skin. Emia involves the blood. Itis is inflammation. Lysis means to cut open, break apart, or destruction. And oma is talking about a tumor. So normal biota is a large, diverse collection of microbes living on and in the body, also known as resident or indigenous biota or normal flora. They include an array of bacteria, fungi, protozoa, and viruses. These organisms have a profound effect on human biology. So acquiring resident biota, the sites that harbor no normal biota, basically anything that opens up to the outside of the body. the skin, respiratory tract and lungs, gastrointestinal tract, outer opening of the urethra, external genitalia, vagina, external ear canal, external eyes, so the lids and conjunctiva, so anything that's open to the outside of the body. Sites that are thought to be sterile include the heart and circulatory system, liver, kidneys, bladder, brain, spinal cord, muscles, bones, ovaries, testes, glands, the sinuses, middle and inner ear, fluids such as blood, urine in the kidneys, ureters, and bladder, cerebrospinal fluid, saliva prior to entering the oral cavity, semen prior to entering the urethra, and amniotic fluid surrounding the embryo and fetus. So basically anything that's internal. Now, a lot of these are being debated currently. So we used to think that the amniotic sac that surrounded the baby produced a sterile environment around the baby. And now recent research is suggesting that no, there's actually microorganisms in there, normal biota, and that normal biota is influencing the development of the fetus. So there's a lot of other things that are going on now with current research. So the benefits of normal biota, they influence the development of organs, they prevent the overgrowth of harmful microorganisms, and then the general antagonistic effect of the good microbes have against intruder microorganisms. If the good guys are well colonized, so you've got a lot of them in the area, there's really no room for the bad guys to bond. And they compete for binding to our inner surfaces. So if the good guys are there, the bad guys are decreased in number or not able to bind. Microbes in a steady established relationship are unlikely to be displaced by incoming microbes. The makeup of your intestinal biota can influence many facets of your overall health. So differences in the gut microbiome have preliminarily been associated with differences in the risk for heart disease, asthma, autism, rheumatoid arthritis, even thoughts, moods, and the propensity for mental illness. This is serious. The type of microorganisms that are living in you and on you are affecting the way that you think. They are affecting the development of autism. They've done all of these studies where they have compared the gut microbiome of children with autism versus children without autism, and they are completely different. So autistic children have this type of microbiota versus non-autistic children. And if we could introduce a certain type of bacteria into an autistic child to help decrease their disability, that would be amazing. But not only this, they're associated with all sorts of immune-stimulating diseases, so like autoimmune diseases. My mom has very severe eczema. It's autoimmune eczema, so her immune system is hyperactive against anything that lands on the surface of her skin. Doctors have suggested using bacteria to help treat that, because bacteria can modulate the immune system. They can control how the immune system responds to stimuli. So it's not just that you've got good guys and it decreases the likelihood of disease. These good guys have a very huge effect on how you act and your overall health. So the initial colonization of the newborn. So the best way for a baby to get colonized is by going through the birth canal. So vaginal delivery is always preferred. When the amniotic sac breaks, all of the fluids from the vagina flush around the baby. And that is a very good source of microbiota for the baby. If a vaginal delivery is not okay or not safe for whatever reason, then the baby's going to be born by C-section, and we want that initial skin-to-skin contact. That initial skin-to-skin contact is going to help colonize the baby. Formula provides added probiotics, so people add in these probiotics while they are formulating the formula that we feed to babies, but the richest source of the appropriate probiotics comes from breastfeeding. Not only from the milk directly, but also from the skin-to-skin contact. So as the baby's mouth goes around the mother's nipple, they're getting contact there, and that's how they can get initially colonized.